Welcome to the Jack Westin MCAT podcast with your hosts, Michael Maleka
and Molly Kilty. Hello, everyone, and welcome back to another episode of our Jack Westin MCAT podcast with your hosts, Mike and Molly. Mike, we've been on a total genetics DNA central dogma kick for the past couple episodes, and I'm excited to really draw a lot of what we were talking about together in our episode today. Gonna be super fun.
And it's hard not to on a topic like this because it's so cool. It's such like a fundamental part of, biology, right? It's really the start point of really all your anatomy, right? It really all starts at the DNA, at the genetics. That's what really kind of decides who we are. But today, it's a really, really cool topic today because we've kind of discussed, we've talked about Mendelian inheritance. We've talked about that, how you get your traits from your parents. We've talked about the central dogma. When you have those DNA, those DNA genes, how do those become the functional proteins? But now we're going to talk about how, while we have this copy of DNA, when our cells divide, how do we actually replicate that DNA? So that is really the focus of today. We're going to be talking about DNA replication and what happens if we have some issues in the DNA. What could go wrong? Well, we're going to be talking about mutations, the cell cycle, cancer. A lot of cool stuff today. I love this topic. So yeah, I'm also, as always, really, really excited.
Yes. Yeah. Today's all about growth, right? If you have one cell becoming two, all of this applies. Right? For whatever reason, they might do that, whether it's a sort of a somatic cell, right? Or if it's a germline cell, if we go through meiosis or mitosis, if we've got any sort of cellular division, any sort of growth going on, right? We need replication. And we've got to figure out what happens if something goes wrong with our replication, right? So we're going to talk about mutations and cancer in here too. Lots of very applicable material. You see this come up quite a bit in our MCAT passages. In fact, we do lots of mutations, especially expect to see that in multiple passages within Biochem within any given exam. Lots of mutations.
Yeah, they love doing like knockouts, mutations. That way we got rid of this. What happens? Because that's really in biochemistry and like in that field, that's how they do experiments. They just knock things out and hey, let's see what happens. And then they put it back in and they're like, what happens now? So being very comfortable with like these knockouts and mutations, super, super critical, especially for passage reading, I feel.
Yeah, most definitely. So in our last episode, we talked a lot about how DNA was used, right? That's the whole idea of the central dogma. We started with DNA that we already had in each cell. And then we talked about how you can transcribe and translate that into RNA and protein, respectively. That being said, every cell in your body has the same genome, little asterisk.
For the most
there are a
part.
couple weird ones, red blood cells, white blood cells. But really, every cell, for the most part, in your body has its own genome. If you have one cell becoming two, you have to make sure that each cell, each daughter cell, ends up with a complete copy of the DNA. Okay, and that's really where we're going to start, and that's replication. So how do we essentially take a copy, make a copy of our DNA double helix, right? So as a refresher on DNA structure, we do have two unique strands of DNA, right? Nucleotides. We talk about directionality from 5 to 3 prime or 3 to 5 prime, right? And it's all about the carbons, right? The numbers we assign to the carbons as part of each nucleotide. So the 3 prime end has an exposed 3 prime OH. The 5 prime end has the 5 prime carbon, right, on the other side. So we have two strands of nucleotides that are anti-parallel to each other. So 5 prime end is next to the 3 prime end of the other strand and vice versa. And then ultimately all these nucleotides are linked by that sugar phosphate backbone. Okay. Keep in mind, those two strands are also complementary to each other. So if you have a C on one side, the G in that same region on the other strand would be a G, right? C and G, A and T form hydrogen bonds that hold those strands together. So want to make sure we're really comfortable with this because if we're trying to create copies, right? And we actually had a ton of really cool DNA experiments that helped us figure out how we replicate. But the real and experimentally proven method of replication, we call it semi-conservative. Basically, we're going to take these two strands that are already present we're going to split them apart and use each strand to generate a new one. Okay. So we are taking one old strand and synthesizing a new one in basically pairing with it. And that's what it means to be semi-conservative. So we have to interrupt the hydrogen bonds that hold the strands together in order to do that and to generate a new strand in that, which is pretty cool.
This idea of like DNA replication, I think really hinges on the fact that DNA is double-stranded. Right? That's so critical because if we only had one strand of DNA, like DNA replication would be much harder, right? But we have this fantastic idea where, yeah, you're right. What you mentioned was perfect, where we have two strands that are complementary. Because a question I get all the time from students is like, wait a minute, I know that we need to separate this idea of semi-conservative. The daughter cells gets one of the old strands and one of the new strands, like a new strand that we make. That's the idea of semi-conservative. One cell has two old strands. The two daughter cells each will get one copy of the old strand and one copy of a new strand. But if they only get one copy of the old strand, and we mentioned that the two strands are not the same, how is it that these daughter cells are identical to one another? Because that's the whole idea, that we create two copies that are identical. And it's the idea of complementariness, I don't know, where if you have one strand of DNA that's A, C, C, for example, when you are creating that second strand, it will be T, G, G, no matter what. That TGG that you created will be the exact same as the strand that you removed. So it's this idea that although you're using one strand as your like template, which we kind of like discussed last episode, that new strand that you create will be the exact same as the strand that went to the other daughter cell, and vice versa. So even though they're both getting only one copy of the old DNA, the new one that they create is going to be identical to the one that went to the other one. So this idea is very important, and this is the idea of being semi-conservative. It's conservative. It has one of the old strands, but not both of the old strands. That's why it's semi-conservative.
Yeah.
And I think this idea is really cool. But if we have this like long strand of DNA, another question people ask is like, okay, so all right, I need to take that old strand and start copying it. Where does that actually start? Now, a common misconception is like, oh, it starts at the ends because those are what are available. And that's not exactly true, right? It happens actually in the middle at these places in the DNA that are really specific called origins of replication, right? Imagine you've got this long double-stranded DNA. It's not at the ends. In the middle, there's these sites that's where the replication happens. And this is where we start kind of breaking those hydrogen bonds, kind of pulling the DNA apart at these origins. And you can actually have on a single DNA multiple origins of replication. So like replication doesn't just happen in the middle of a strand and then just goes out to the ends. No, in fact, there's multiple kind of origins of replication that will all kind of meet together. And so this is kind of where the story of replication starts, at the origin. And now I think this is kind of a good place to talk about what key players are actually involved at these origins, what are actually like the order that this actually happens, because there's a whole process. This isn't just random. And I remember when we talked about last episode, we were talking about transcription and translation, especially transcription. We were like, there's RNA polymerase, that's it. It was like, that's the start of the show. Like, that's all you really need to focus on, along with a few transcription factors. But, we weren't so specific on that. But when it comes to DNA replication, there's a lot of players here. And it's important to know the order that they come in, Molly, right?
Yes, most definitely. Yeah, we had it easy in transcription, whether
Right.
you knew it or not, because RNA polymerase can do pretty much all of these jobs for itself. We also, though, had an easier time because ultimately we had two strands, but only one of them had the sequence that the RNA polymerase was scanning for, and we took one of those strands and made one strand of mRNA and called it a day, right? That was it. We just had to synthesize one strand. In this case, we have to separate DNA, and then we're making two strands. Each one of the original strands of DNA becomes a template for the synthesis of a new one. So we've got a couple new layers of complexity that we're introducing, and ultimately we've got many more enzymes that we're going to discuss. Okay, so let's start at the beginning. We have two strands of DNA, right, that are bound together by hydrogen bonds with each of those base pairs, okay? like you said, we have origins of replication, and there are actually multiple along the long linear sequence of DNA. Keep in mind, if you're curious with prokaryotes, a lot of this is the same, but a prokaryote has one circular chromosome. So there's actually only one origin or replication. And again, it's a circle instead of a linear chromosome. But we're going to grab essentially onto those two different strands of DNA, and we're going to start pulling them apart. Okay. This is primarily done by an enzyme called helicase, okay, that ultimately is unzipping those two strands. And this is a great process, right? Helicase is literally functioning like a zipper, right? It's moving into the double helix and creating separation. It's disrupting those hydrogen bonds. But if helicase just moved right through, you could almost imagine that the DNA strand would be zipping itself back up after helicase continues to move, right? So when we disrupt those hydrogen bonds, that's great, but we don't want them to immediately reform. So we have helicase come in, and then we have little baby proteins called single-stranded binding proteins that are actually going to come in and satisfy the desire for those nitrogenous bases to form hydrogen bonds and essentially keep them held apart. Okay, so helicase comes in and unzips. Single-stranded binding proteins are going to stabilize them when they're separated, so they don't immediately come right back.
Right.
And when they do this, just so you know, there's another enzyme that's often brought up in this unzipping process called topoisomerase. And really, topoisomerase relieves what we call supercoiling when we do this. Okay, basically, if DNA has a double-stranded double helix, I've got a little model.
Right, love that.
Right, they are wound around each other, right? And when you come in and you start to just yank them apart, it actually takes this normal, like sort of winding pattern, and it just concentrates it on either side. The classical description of what would happen was usually historically given with phone cords. Like, oh, you've got a phone cord, and if you
really?
if you take your finger in your phone cord and you like pull it down, it creates like all kinds of weird knots in the phone cord.
Yeah.
But nowadays, every phone is cordless, and people don't have.
They're like, oh, how do I visualize
Yeah,
that?
and then it became headphone cords, but most people have wireless headphones. So for me, the way that it makes sense, if anyone has a rubber band or like a hair tie, if you were to twist at both ends, you start to create all of these, again, little twists, little helices, but as you continue to do so, they create these weird three-dimensional little loops and coils. Like it doesn't want to just twist and twist and twist forever. You find that it bunches up on itself in this extra weird shape. This is what happens with your DNA. And
Without
this is very dangerous.
Yeah.
Yes, you don't want this. This is going to break your DNA. Right. If you just kept twisting and twisting and twisting on your hair tie, it's going to snap eventually. Too much tension. So topoisomerase's job is to come in and actually cut the DNA, unwind it, and then join it right back. Right. So it prevents this buildup of tension as you just twist and twist and twist from multiple sides that creates these weird supercoils.
So I love those examples.
It's super cool.
It's so funny because again, I think we mentioned this a long, long time ago. But yeah, Molly and I always had like different how we learned this was so different. And I remember so vividly learning this for the first time, like with just a normal rope, like like a hemp rope, like because a hemp rope, it's two things coiled together like DNA is, and you can imagine as long as the ends are affixed. Like they're not like free, which most ropes aren't. If you start trying to pull a rope apart, like you can imagine if you just try to pull apart, the ends are going to start like getting all tied. I remember my professor actually brought a rope to class. It was like, let's see what happens when I pull these two things apart. And I remember visually saying,
I love that.
The ends actually coiled on their own. And so that's how I always thought about like super coiling. It's like, it's just a rope. If you pull the rope, yeah, you can imagine like the ends are going to get all tied. And that's what, again, it's so dangerous for that to happen because then now you're going to cause DNA damage. Now you can't even replicate those anymore. Like imagine for, all those proteins to get in there. But that's what like topoisomerase do. And he actually cut his rope. actually cut like little snippets in the rope to show what topoisomerase does, because by making those little snips, those little cuts, you relieve all that tension. So that's how I
How
so
cool.
see,
I love that.
I never did like the phone thing. I was like, oh, I'm like, oh, that's a cool example. I never thought about that. I always just thought of it like a rope. But yeah, so hopefully, as you've been listening, right, we've talked about three proteins so far. Helicase, what actually initiates, there's at the origin of replication, actually starts the unwinding. SSBP, single-stranded binding proteins, making sure it doesn't just zip back up with those hydrogen bonds. And then topoisomerase, relieving that supercoiling on the end so your DNA doesn't just wind together. But we haven't actually started replicating it, anything,
Oh,
have
we just held them apart.
now they're just apart. And now, are we actually ready to get like started? Can like our kind of action actually happen quite yet?
Well, not really.
Not really. Hmm.
Kind of, but not the DNA synthesis
Hmm.
yet.
Hmm.
DNA is not so good at being a trendsetter.
I love how you not very trendsetter DNA.
Yeah.
RNA can happen on its own.
RNA will take its own initiative and will do
Right.
its own thing. Keep in mind, RNA polymerase has no problem just striking off on
Binding and starting.
itself, but yeah, DNA not so much.
I love that we know what we're talking about, but it's like everyone's like, what are you guys talking about? So for DNA replication, right? So our major kind of like action protein in RNA, for RNA transcription, it was RNA polymerase. In DNA replication, it's DNA polymerase 3. That's the main star. But a big difference between the two is DNA polymerase can't build off nothing. It can't just start building. It needs a sort of like start point or something to build off of.
Scaffold.
Yeah, kind of a scaffold, right? I was actually gonna be like, oh, when you're building a house, you need scaffolding to build around it. Thanks, Molly. I'm just kidding.
I ruined your punchline.
It needs a scaffold to build from. RNA polymerase didn't need that. DNA polymerase 3 does. So that introduces our next key player, which is, I've heard both DNA and RNA primase. I've heard both of them. I always learn it as RNA primase to help remember what that primase does. Because what RNA primase does is now we have this open origin, this kind of replication bubble, we call it. That bubble is kind of open now. And what RNA primase does is it goes and it just places one RNA, one like nucleotide there, right? RNA nucleotide there to kind of get things started. Because one RNA primase, like actually, it's not even just one, it's really just a primer. We call it an RNA primer. It's like a couple, it's like a very, very small piece. It places this RNA primer down. And once that primase actually places the RNA primer down, now you've started the scaffolding. You've started kind of things to go. And so now our next key player can come in. And that is DNA polymerase 3. It is very important. Here's another big difference from RNA polymerase. RNA polymerase was just RNA polymerase. There's no numbers involved. There's just one. DNA polymerase, on the other hand, there's like 4 different DNA polymerases. However, for the MCAT only really focuses on 2. The first one that it focuses on is the main key player, which is DNA polymerase 3. And that's the one that's really the big workhorse of the DNA, of DNA replication, where it will bind, it will start at that RNA primer. Again, I want to really emphasize that this is RNA that it's starting at, and it will start building. Now, super key, which was exactly like transcription that we talked about last episode. You can only build in the 5' to 3' direction. Only. Always, that's always the case. And therefore, in order to build 5' to 3', you must be reading 3' to 5'. So when that DNA polymerase 3 binds, it's going to start building 5' to 3' and reading 3' to 5'. But wait a minute, Molly.
And
something's a problem. This is an issue here.
yes, there is an issue. And just to clarify language here, when we say reading, keep in mind DNA polymerase is synthesizing the new strand. So it's actually facilitating the building of a sugar phosphate backbone. But these strands have to be anti-parallel to each other and they have to be complementary. So we have a new nucleotide that's coming in, right? That should properly form, it should form the right base pair, right?
Sure.
A to T, G's
Should
to
be.
C's. And then, yeah, it should be. We'll talk about mutations and issues with that. DNA polymerase is making sure the right one is there and then helping take that nucleotide and add it to that growing chain. Okay, so DNA polymerase 3 can't add to nothing. It has to have a 3' OH, right? Because that's how it's going to attach the next nucleotide. So it's making the new strand in a 5 to 3 direction, right? But in doing so, it has to be reading the old strand. Okay, the reason why we synthesize 5 to 3 and we read 3 to 5 is because the DNA strand is anti-parallel. Imagine just going from, let's say, left to right. You're looking at the new strand from left to right. You have 5' on the left, 3' on the right. But if you're looking at the one that you're reading, it's going to be the opposite direction. Okay, so the enzyme itself is moving in one direction, but because those are anti-parallel, the direction is different. Okay,
Right.
Students always get confused about what does that mean to read versus synthesize.
read versus
Two
synthesize.
opposite strands, the strand that's getting made versus the strand that's already there. That's the template, right? But you're totally right. If we have a replication bubble, we pop it open, right? And we now have helicase moving off into where DNA is still present in a double
Unraveling.
helix, right? We're moving off, let's say, from left to right. Okay, we've got, let's say, the 5' end of the top strand on the left and the 3' end of the bottom strand on the right. Okay, for the bottom strand, right, if we're moving from left to right, we're going to go from 3 to 5. This is the original DNA strand. That's fine, right? Because we can
good, no problem.
synthesize 5 to 3. That's the proper direction. We're going to lay down one little primer there in the replication fork. And then DNA pol 3 can just keep hanging out. As helicase breaks apart those strands, right, the bottom that's going from 3 to 5 in the direction of the replication fork opening, it's happy. It could just keep synthesizing until it beats the next replication bubble and it's happy and it's done. The problem is the top strand because it is 5' on the left and it's opening towards the 3' end on the right. Now, DNA polymerase 3 has to read 3 to 5, which means like my replication bubble is on the far left side of your imagination. And my replication bubble
or picture on the podcast if there is you want to put a
we
picture.
have a picture. And we're extending to the right. I've got to find a spot towards the right-hand side of your imagination, lay down a little RNA primer, and then I've got to extend from right to left. I'm then going to reach the end of my imagination, and my helicase is going to have opened up farther to the right, which means I'm going to have to add another RNA primer and then extend from right to left again. And then I'm going to run into my old DNA. And then I'm going to have to go in further, add another RNA primer, extend until I reach my original DNA, and just keep going in pieces as the replication fork opens. This is what is complicated, right? With transcription, we were able to separate and we were only making one strand. Great.
Yeah, easy piece.
It's easy. Everything is what we call the leading strand, right? The leading strand is the one that just gets one RNA primer and then it just extends and it's easy. It's beautiful. It's gracious. Like it's just
beautiful.
it's,
It's gracious. It's kind. We love it.
It's simple, right? Versus the poor lagging strand, in order to keep up with the replication fork, has to be synthesized in so many little pieces. And so when we are done, right, we're going to end up with all these little fragments. One thing though that I want to add, because I think it's really, really cool. People are always like, oh, that stinks for the lagging strand. That's awful. Fun fact for you, each of the strands, what originally was the top and the bottom, when we pull them apart, right, those two original strands actually will both be the leading and the lagging.
Yeah.
The top strand, I said, was the lagging when we're going from left to right. But keep in mind, replication forks or replication bubbles actually open in both directions at the same time. So we have a opening and then we're moving from left to right. And at the same time, we're moving from right to left. So what's leading on one direction of the replication bubble moving is actually lagging on the other side and vice versa. So each DNA strand that's synthesized will be an equal amount leading and lagging.
Maybe not equal, but like it will be a mix of like
it'll be proportional. Yeah.
Yeah, it'll be a mix. Yeah.
It depends on where your origins of replication are, but yeah, so it's kind of cool. You don't have one strand that is
That's just
leading the whole
all,
way,
yeah.
and one strand that is lagging the whole way, which is
hmm
pretty cool.
It
I
is
love that.
so, and I love that you were like, poor upper strand or poor low. I love how kind everyone is to the DNA. But yeah, and these fragments, I don't know if we mentioned it. These fragments actually have a name that we should know, and it's actually
Hmm.
named after the researcher that discovered these. So these are called Okazaki fragments. So all of these little pieces that we're building, right, that are all like really separate because it's not continuous. They're all like we're like starting and stopping, starting and stopping, starting and stopping. They're all fragments. They're called the Okazaki fragments. But now, all right, so imagine maybe we've done this all. Like we've had all our bubbles, right? All our bubbles happen, and maybe our bubbles met now, and now we've synthesized the DNA. But all right, are we done, Molly? Like, is it like all right, we're good to go? We're like, all right, time to separate everything now.
No, we've got some issues.
Lots of issues right now because we've got like two main issues in my mind. One, this is supposed to be DNA, but we have a whole bunch of RNA in our
Hmm.
DNA. Because remember, those primers that we set down, because remember, DNA polymerase 3 has to build off something. Those were all RNA primers. We cannot be leaving RNA in our DNA, right?
Hmm.
Problem number two is our lagging strand, which is we've got a whole bunch of fragments. We need a continuous strand of DNA, not just a whole bunch of fragments. And so that's where two really important key players comes in. The first is what we call DNA polymerase 1. Remember, we had DNA polymerase 3. This is like his younger brother, DNA polymerase 1, or older brother. I kind of like think like DNA polymerase 3 is kind of like the younger brother that's kind of just going and doing stuff. But DNA polymerase 1 in my mind is like the older brother who's like, okay, now I've got to fix some things, right? Like my little brother was doing. As you can see, I'm an older brother. Let me fix what he did because what DNA polymerase 1 does is it takes all these RNA fragments and essentially gets rid of them. It sort of chews them up via it's and this direction I want to make sure is right. 5 prime to 3 prime exonuclease activity. Yeah, 5 prime to 3 prime, where it goes to that open end of the RNA primer and chews it up. And in the process, replaces it with DNA. And it puts the DNA in that 5 prime to 3 prime direction. So it has that exonuclease activity and then actually replaces it with the DNA. So that's problem number one solved. Now, DNA polymerase 1 is going to go and chew up these RNA fragments and actually make DNA there. But problem number two is still there. Well, they chewed up the RNA and now there's DNA, but we still have a bunch of fragments everywhere, even if they're DNA now, but they're still broken apart. And that's when I think this enzyme is actually really common. I think people know of this enzyme when they learn about it. It like makes sense. And that's the kind of the key ligase. Remember we talked about enzyme classes? Ligase is the one. This is where DNA ligase comes in. And DNA ligase, really simple job. Just whatever the fragments are, stick them together. Right? Reform that phosphate backbone that connects all those nucleotides together so that we have one continuous strand of DNA. On the leading strand, all it needs to do is at that first place with whatever was there. But on the lagging strand, it's got a lot of work to do to connect all those fragments together. But that's now, through the action of DNA polymerase 1, and we got our DNA ligase, now all those problems are solved. And now, because this action happened on both the upper and lower strand in both directions, we have, once we finish all of this, we now have two hopefully identical double-stranded DNAs, right? And now we're done with replication.
Hooray. I
Woo!
love that you say hopefully identical because
Hopefully.
ultimately we'll talk about cell cycle and the
hmm
different phases of the cell cycle, right? This takes place when we're getting ready to divide,
hmm
right? That's the goal is I'm one cell that wants to become two. I need to make sure I have two copies of DNA so each daughter cell will get one, right? That's the whole point. So we're going to pick up from this point in cell cycle, but first, we're only hoping that these are identical
Hopefully.
because
Yeah.
sometimes they're not, right? We talk about DNA Pol 3 as the workhorse and it's doing so much for us and it is, but it's not perfect,
hmm
right? Sometimes while DNA Pol 3 is working, sometimes it makes a mistake.
Mm,
Sometimes it puts the wrong nucleotide there.
mm,
And sometimes it's really obvious and sometimes it's not. And it kind of slips past some of our surveillance systems, right?
mm,
We'll talk about different layers that we proofread. In fact, DNA Pol 1 and DNA Pol 3 both have the ability when they are looking at the new nucleotides that they added in, right away they could say, hold on, whoa, wait.
Let's take a
I
look.
think that was wrong.
Yeah.
Right? You mentioned before when you cut out RNA primers, it's 5-3 exonuclease. I like to think that's the direction that they're looking. They're starting at the 5' end and looking towards the 3' end. The 5' end had the primers, right? Because you extend 5-3. This is 3-5. You're at the 3' end looking towards the 5' end. So you're looking at the nucleotide you just added. Pol 1 and Pol 3 can both, if they realize, oh shoot, shucks, right? I added the wrong nucleotide. They could fix it right then and there. Great. No mutation, right? Because you proofread it, recognize that you did something wrong, and you fix it. That being said, things get past our replication machinery. Not super often, hopefully, but it's not abnormal, right? And typically what we see is when you change DNA structure, you then mess with the amino acid sequence or the RNA sequence that could be made from that DNA template. Sometimes these mutations are really minor and sometimes it doesn't even change anything, right? But sometimes these mutations can be incredibly impactful.
Huge
so
impact.
When we talk about mutations, there's really two main levels that we discuss with actual replication issues. There's a point mutation and there's a frameshift mutation. Now, a point mutation is typically going to impact just one amino acid. That's it, right? So I've changed one nucleotide in my DNA, which led to a change in the nucleotide in my mRNA, which led to something that affects one amino acid, okay? We have a couple different types of point mutations. We have a silent, right, which is exactly what it sounds like. You have made a change in your DNA, but it actually doesn't change the amino acid that gets placed there at all. So your codon in the mRNA that was made from your DNA is slightly different, but it's close enough that it's actually the same amino acid that it codes for.
Right.
So it doesn't really do anything. You changed the DNA, but it didn't impact the protein whatsoever, right? It's the same amino acid. It folds the same way, same activity. That's why they call it silent, right? A missense is, okay, I changed the nucleotide in my codon. Let's say it codes for a different amino acid, okay? Maybe I went from leucine to aspartate or something like that, right? I changed one nucleotide and it actually created a different amino acid. We then have to wonder, okay, it's just one amino acid in a large protein. How significant was that, right? And that's when we start to think, what was the original protein or the original amino acid that was supposed to be there? What is the next one, right? I said leucine to aspartate. Leucine is nonpolar. Aspartate is negatively charged. That might be a little disruptive, right? If it was leucine to, let's say, isoleucine, probably not going to do anything. They're both nonpolar, very similarly sized, right? So then with missense, we need to wonder, okay, I've replaced an amino acid. How different were they, right? Is that really going to impact the structure of the protein and the function of the protein? It depends, right, on the properties of the amino acids. The third type is typically the most disruptive of all of them. Although, again, all of these depend on context, right? What was switched, where in the DNA sequence was this occurring? The last point mutation is called a nonsense mutation. And in this case, we change the DNA such that the RNA nucleotide or the RNA codon that is read by the ribosome during translation actually now, instead of coding for an amino acid, is a stop codon. So it's one of our UAA, UAG, or UGA. We now have a stop codon that's way earlier than it was supposed to be, which means that it's going to create what we call a truncated protein. The protein is going to stop too early. It's going to be too short. And typically, this is pretty problematic. If your amino acid is supposed to be 100, or if your protein is supposed to be 100 amino acids, and you have a nonsense mutation at position 99, so you have a stop codon one amino acid earlier than you should, probably not going to be the biggest deal, right? Like your protein's almost done, but imagine the second or third amino acid in your protein that's supposed to be 100 was a stop codon, right? Like context matters. Where is this happening?
So much.
Yeah. So these are all our point mutations. Again, we are just changing one amino acid. There's silence, there's missense, there's nonsense. If we suddenly add or delete nucleotides instead of just putting the wrong one in a given place, now we're a little more concerned, right? We call
Mm,
this a frameshift mutation because what it does is it shifts the reading frame of, right, actual translation. Keep in mind codons, which is RNA, these are read 3 nucleotides at a time. Okay, if I suddenly add an extra nucleotide, that becomes, let's say, the first one that is read, and it'll take the first 2 of what would be the next codon, and it shifts the whole frame over by 1 nucleotide.
mhm.
Now, every single amino acid after that insertion is different because I've shifted the reading frame. Okay, deletions are the same deal. That being said, not all insertions and deletions are problematic.
Mm,
When might we expect that a
mm.
frame shift, or you could insert or delete and not shift the reading frame?
That's a tricky question, and one I've definitely seen where it's because it's like, okay, if I add one or delete one, it will shift the frame. If I add two or delete two, it will shift the frame. But if I add three or delete three, the frame's still the same. It's just maybe I add one amino acid or I get rid of one amino acid. Doesn't mean it can't be absolutely problematic for the protein. But if we're talking about comparisons now, additions or deletions of three actually aren't generally not as bad because the frame is maintained. It still reads the same three bases for the codon. Is just now we've now added one, or now there's one less is the key. So that's kind of the
Mhm.
one place where additions, deletions might not actually shift the frame.
Yes.
Yeah.
Yeah, and that's so weird. And keep in mind if you added 6 or
Mhm.
deleted 6 or you added 9 or deleted
Sure.
9, it's the
Mhm.
same deal.
Mhm.
Frame shift mutations shift the reading frame.
Mhm.
But if you change the sequence in a multiple of 3, it doesn't shift the reading frame.
Mhm.
Like you said, you'll add or delete amino acids, but that's it,
Mhm.
right? So again, there have been a lot of questions in AEMC material about which of the following is like the most impactful, which
Mhm.
mutation is the most impactful for a protein. And you might see like a silent mutation. You might see a
Mhm.
missense mutation that's nonpolar to nonpolar. You might see a frame shift mutation at the very, very end
Mhm.
of a DNA sequence, right? In terms of where it's read for the protein. And then you might see a nonsense at the beginning. Which one would be most consequential in an option like that?
Nonsense, 1,000%. Nonsense at the beginning, because you might see that in the answer options, and you're like, wait a minute, frame shift and nonsense. They said
Those are both bad.
both of them can be really bad, but where it happens is going to be
Hmm.
critical. Yeah, if it's over at the end, yeah, it might change a little bit at the end. Not to say that that can't be something bad, but yeah, something like a nonsense, like you turn like 100 amino acid to like two, now that's a really big
That's a
problem.
problem.
But
Yes.
doesn't mean, right? Like I said, context dependent. A great example I always like to think of is actually sickle cell anemia is actually a point
Hmm.
mutation. That's a missense mutation of one amino acid difference. And yet,
Yes.
sickle cell anemia has huge impacts on the body.
Yes.
So I always like thinking that as context to say, like, just because it's not um, nonsense or frame shift doesn't mean it can't have huge impacts. But if we're talking about likelihood, those are more likely to have bigger impacts.
Yes,
Yeah.
for sure.
Mhm.
So these have all been differences just in the replication process,
Mhm.
right? There are other issues that we can have with chromosomes as a whole,
Mhm.
right? So this is not just with individual nucleotides while we're polymerizing, right? There are also chromosomal abnormalities. We've talked about non-disjunction before, when you accidentally bring both chromosomes into the same basically both copies of a chromosome into the same daughter cell, right?
Mhm.
So you have one daughter cell that has
And
uh,
one.
too much genetic information and one that doesn't have enough, right? There's also translocations where you can have parts of chromosomes basically switch places or
Mhm.
part of one chromosome gets added to the next. These are not typically done during replication, but they are also problems that can arise when we're looking at our DNA, right? So
Mhm.
they're valuable to bring up, but in terms of like our actual replication machinery, these are the things that we most often talk about.
Yeah, absolutely. And so I think we talk about like these errors when it comes to replication, but there might actually also be other reasons we can have mutations, right? Because DNA
Mhm.
replication, like we said, that could be, where our DNA poly is going on, or DNA like what, three or one. But source of mutations can also be things like that are environmental. A very common one that we talk about is like sunburn, right? A question that I always like asking students is like, why do you think like skin peels when you get a sunburn, right? Like we've had it, and actually I was on vacation recently and I got a wicked burn. Would not recommend. Please wear your sunscreen.
Me
But
too, actually.
so I was in the sun. It's because I was reading and I fell asleep. Like I got a wicked sunburn and my skin started peeling. But now the question is like, why? Right? And what actually
Mm.
happened is due to the UV radiation. Now, UV, I don't think we've actually talked about like wavelengths or light energy at all on the podcast, but UV is a higher energy wavelength of light, right? Where it can actually cause mutations in your DNA, specifically something called a thymine dimer, where you and your DNA, right? You've got A, C's, T's, and G's, right? You've got the four nucleotides, or the bases. But let's say you've got two T's next to each other. It happens all the time. With the exposure of intense UV, what can actually happen is they can actually form bonds to each other, right? That are next to each other on the same like strand of your DNA. Normally, they should be bonding to the opposite, to the complementary strand. T's should be bonding to their A's, right? But then they start bonding to each other and form what we call a thymine dimer. This causes a little bit of a kink where if it's not dealt with, can actually cause a lot of damage to the cell. And your skin peels because those cells, they tried fixing it, but there's so much damage that they just die. They apoptose because of the extent of the damage. Now, we're going to bring in a few more pieces of context there, but that's a really great example of times when you can have a mutation that's not due to DNA replication. There's other causes of these mutations, but I kind of hit on something that we kind of already mentioned before about when there is damage, how do we deal with it, right? You mentioned before, Molly, that sometimes our DNA polymerase can kind of catch their own mistakes, right? That's kind of like
Mhm.
the first kind of level of protection, right? Which we call proofreading, where, like we said, we build 5 prime to 3 prime, but maybe they actually put a base down. Maybe they on the strand that they're reading, they see an A. They're like, okay, there's an A there. But instead of putting a T, that they should be building a T, maybe they put a G instead, a guanine instead. Well, if they put that guanine down, there actually will be an issue because now the DNA is going to be a little bit more bulged or contracted than it should be. And so it could see that and like, oops, I put the wrong one down and can actually exonuclease, right? When we say the term nuclease, that means to kind of digest nucleotides. And by exonuclease, that means it's digesting from the end. And the direction, like you said, where you're looking, because we're building on the 3 prime end, we're kind of deleting one. So we're going from the 3 prime to the 5 prime. And that's kind of the first kind of repair mechanism where we can see damage, we can delete one because we put the wrong one down, and then just keep going with the right one. Hopefully that's all. But let's say this is after, where now, we're post replication, but now when we're checking the DNA, we're like, oh, wait a minute. Wait a minute. There was a base that was put there that is mismatched. That there's a base that's kind of a mismatch there that the DNA polymerase put it down and shouldn't be there. That's what brings up our second repair mechanism, which is our mismatch repair, right? And this is post replication, right? But really soon after post. Like it's not very long. It's very soon after because we check after we synthesize the DNA. And in mismatch repair, we see the mismatch. And again, how they see the mismatch is still via like bulging. Like imagine if I put two purines together, because normally when we do DNA complements, it's always purine pyrimidine. So there should be 3 rings. For context, before let me take a step back. Purines have 2 rings, pyrimidines have just 1 ring. Because DNA is complementary, a purine should always bind to a pyrimidine. And so therefore there should always be 3 rings in the middle of the helix. And that should always be the case. And then therefore the DNA will be uniform in distance from the 2 strands should be uniform. But if I match 2 purines or 2 pyrimidines together, if I match 2 purines together, now my DNA is going to be too bulged. If I add 2 pyrimidines together, now my DNA is going to be too close together. And our detectors, we have little like damage detectors in our DNA, look for these bulges and areas of thinness. And that's how they say, oh, hey, there's a mismatch there. And in mismatch, what they're going to do is they're going to clip the whole region, right? They're not so targeted. They're like, oh, let me just get rid of that one. Not how it works, unfortunately. What they're actually going to do is they're going to be like, okay, that one is the problem. Everything on the left and right side, not everything, I should say, a few nucleotides on the left and right side, we're going to cut out and we're going to get DNA polymerase back and remake that region of DNA, right? It's kind of like cuts it out and then remakes it. A question I get all the time is how does it know which is the right one, right? How does it know? Like, say you got two T's together. How do you know which is the wrong one about the T's? And actually, I do know the answer to this, Molly.
Huh?
Do you know the
How do I
answer?
know which one is wrong?
Which one's wrong?
No, tell
Yeah,
me.
it's again. So this was my research when I was in undergrad. So again, I was really interested in this because when you have older DNA, it's more methylated. We actually put methyl tags to know which one's the old, which one's the new one. And so the one with the methylations, that's how we know. Oh, that's the original one. That means that's more likely the correct one. So the one without all the like methyl tags is the one that it actually replaces, which I think is really, really cool. And how the body actually distinguishes what's the one that was the original and what was the new strand that we need to fix. So very, very cool on mismatch repair.
Sweet. Yeah.
Yeah.
Mismatch repair. So we had a problem, cut out the region, replace it after
Mhm.
replication.
Mhm.
Now, when you were talking about UV damage, if
Mhm.
especially this is the most common case, if you have one of those thymine dimers,
Mhm.
right? Meaning two thymine nucleotides have basically when next to each other on the same strand have come together and formed a dimer. They actually break the hydrogen bonding
Mhm.
pattern of DNA that it would normally have with A's and T's because now they're interacting with each other, right? This is a problem. And like you said, if this accumulates to a really, um, you know, extreme extent, the cell may just undergo apoptosis. That
Mhm.
being said, if you get a mild sunburn,
Mhm.
right, something that's very small, something very
Mhm.
minor, you can actually fix this. And we call
Mhm.
this nucleotide excision repair, right? And this has to do with a damaged nucleotide. It's not mismatched, it's actually damage to the nucleotide itself because of this, in this case, thymine dimer, right? And again, when we have two thymines coming together and it disrupts that hydrogen bonding pattern, this also creates this weird little bulge on
Mhm.
one side only with those thymines that are kind of coming together and creating this little kink where they're
Mhm.
also not held to the other strand. Okay. And this is a very similar response. We're going to come in, we're going to cut it out and its neighbors, nearby neighbors, and we will fix it, right? We'll have
Mhm.
to come in, add new nucleotides. And then again, I don't know if we mentioned this, you need ligase to come seal it up.
Mhm.
Again,
Mhm.
you'll always need that to make sure that you have no gaps in your sugar phosphate backbone. And this is really important because if we don't catch it, and this is true of all types of DNA damage, if you don't catch it and you let that damaged DNA go on to, populate a new cell and then that cell divides, if you think about what would happen if a DNA pol gets to this thymine dimer, it's like, what am I supposed to add here? Like it
Mhm.
doesn't recognize thymine. It doesn't have that ability to hydrogen bond when those two have come together. So DNA Paul's like, huh? And
Mhm.
it pretty much just adds whatever it wants in that little gap and then keeps going. So this is a really important source of
Mhm.
mutation, and that's partially how you get skin cancer as a
Mhm.
response to extreme UV
Mhm.
exposure, right? Or
Mhm.
certain types of skin cancer. There are actually
Mhm.
many different types,
Mhm.
but that's very common, and it tends to be present in the external site or external layer of your skin. But again, nucleotide excision repair, there's something wrong with my nucleotide itself.
Mhm.
I need to go and fix it. Okay. And it tends to involve multiple nucleotides. Now, there's also what we call base excision repair. And this is when we have one base that has a problem. Specifically, this is most often with cytosine.
Mhm.
Cytosine
Well, problematic
is
guy, that guy.
that problematic cytosine. So cytosine is very similar in structure to uracil, which
Mhm.
is an RNA. Um, it's an RNA nucleotide, right? But it's only ever found in RNA.
Mhm.
Okay. If we see uracil, that base in DNA, we get very confused. It should not be there. Okay. If cytosine is deaminated, it actually has a nitrogen containing group instead of a like ketone carbonyl group. If cytosine undergoes deamination, this is damage to one base, just the nitrogenous base, nothing else is changing. We can also go in and fix that. This tends to be a lot more directed. Okay, we'll go in, we'll cut the base out, we'll cut the nucleotide out, and we'll replace it with the new correct nucleotide. So this is a lot more targeted because it's not an issue with entire nucleotides. It's only an issue with my base is damaged, right? So we're going to go in, it's much more specific, cut it out, replace it, and call it a day. But again, you don't want to see uracil in your DNA. That's a problem. And again, when you go through replication again, not only is your DNA pool going to be really confused, but it'll probably put an A there, if anything, right, to match with uracil. On the other side, though, C, which ultimately became U through deamination, was paired with G. Okay, so when that strand is read, G is going to be paired with C. So now I have in my two different strands, one A that was added and one C. That's the problem. And again, all of these mutations individually, typically not that consequential. You get one and it's like, okay, it's normal, right? Mutations accumulate as you live your life. It's not typically a major problem for one to happen, but it actually really depends, right? What did I mutate and how significant of a mutation was it? How important is that protein or mRNA product that I need? Like, is it critical? What cell type is primarily using that gene? Is it something that's present in everything, right? How important is this? When we have mutations accumulate, this is when we start worrying about cancer, right? Because adding together a bunch of mutations, right? You get one and it's almost like a domino effect. It makes it a little easier to get a second one and a little easier to get a third. You reach a point where you have accumulated so many mutations that it's like a free for all, right? Your DNA is doing whatever it wants and you've got very, very abnormal cell behavior. So in our process of getting into that, right, we got to understand the cell cycle. Really, cancer is dysregulated cell cycle. It's just dividing and dividing and dividing, but we need to understand why. So we got to talk about the cell cycle.
We got to talk about the cell cycle. And I think what we really want to focus on the cell cycle is where the DNA gets checked. So as an overview, the cell cycle has kind of four main phases in active replication, right? And we'll talk about the fifth phase if we're not actively replicating. So the first phase is known as the gap 1 phase or the G1 phase, not the growth 1 phase. I always love bringing that up. It's not growth phase 1, it's gap phase 1. And here, this is the kind of the first quote unquote stage. And this is where the cell's like, okay, I'm going to be replicating. I need to start growing, right? So here, imagine the cell doesn't just split its DNA and replicates DNA, but it's also replicating like all of its cellular contents, like all its cytoplasmic contents, the mitochondria, all these things we need to start producing more of those if I'm going to share it between two cells. So in G1 phase, like is kind of where we see a lot of growth, a lot of like, okay, we're making a lot of things and we're also getting ready for DNA synthesis because that is the next phase, which is the S phase. It's the synthesis phase. And that DNA replication process that we just discussed quite in depth happens during this phase. So before it was just one DNA, but now we've got two copies, two double-stranded helices that are now ready to kind of be separated. That's an S phase. After the S phase, there's the G2 phase or the gap 2 phase. And this is where, again, there's more growth. We're still like adding more things. We're still getting a little bit bigger. But here's where we're really preparing for mitosis. And that's the next phase. So it goes G1, S, G2, and then mitosis. And now mitosis and cytokinesis, because technically cytokinesis isn't part of mitosis, right, where this is where we're really focusing on separating the DNA, right? Like we created two copies and now we need to partition it to two different cells. That's what mitosis is focusing on. And then we have cytokinesis, which separates everything else, right? Where it's just like, okay, mitosis focuses on separating the stuff from the nucleus. Cytokinesis, everything else needs to be partitioned. The mitochondria, the ribosomes, all those extra stuff will be partitioned. And so then after mitosis, you now have two, mitosis and cytokinesis, you now have two separate cells. And then you start the cycle all over again. That's why it's a cycle. It is a circle. You go G1, S, G2, M, mitosis phase, and then back to G1. Now, the fifth phase that I kind of want to mention is maybe I'm not actively replicating. There is a phase called G0 phase, and that's kind of like a offshoot of G1 where if I'm not actively replicating, I'm in the G naught phase where I'm just kind of chilling. I'm not actively replicating. I'm fine kind of deal.
Or G0 for us Americans.
Oh, yes. Sorry, my bad. Yes, it was G0.
Yes.
Yeah.
And I feel like that's important to bring up because there are certain types of cells in our body that really don't divide that much, right? Think your neurons. That's why you need to wear a helmet if you're doing something dangerous, right? Like protect your neurons. They really don't divide via mitosis very often. Same thing with your muscles, right? Even, you know, if you think of like the buffest bodybuilder, right? They don't have that many more muscle. Yeah, like Mike.
Stop laughing.
Mike doesn't. You brought it up. No one, you know, most people have pretty similar numbers of actual myocytes, muscle cells, and yet some individuals have much larger myocytes than others, right? The actual cells get bigger. They call it hypertrophy, right? Not growth in number, but growth in physical size, right? So muscle cells are also a type of tissue that don't divide by mitosis that much, right? There are lots of cells like this. And then, of course, we could talk about things like stem cells, which just divide, like almost constantly, right? Especially those in like our bone marrow or in our skin, right? Like hair cells. Yeah. Like there's certain cell types go through this process almost constantly, and some almost never do it. So it's important to talk about things like G0 or G naught. And ultimately, when we talk about other types of cells, like our germline cells, they go through this same process, but instead of mitosis, they can undergo meiosis, right? And we're not going to talk a ton, we're not going to walk through every step of mitosis and meiosis, but just so you know, it's the same lead up, but rather than M being mitosis, M can be meiosis. And the key difference with the germ cell, you have one regular, I'm putting this in air quotes, regular cell that has a normal amount of DNA. You replicate that DNA once, right? And normally you replicate it once and you divide once and you call it a day. Each cell, each daughter cell has the same amount of DNA. The thing with meiosis though, is we replicate it once, one S phase, but then we divide twice. So we make 4 daughter cells, right? With only twice as much as DNA as normal to start with. So each daughter cell, which is a gamete, right, ends up with half of the normal amount of DNA. And that's how you have fertilization, right? The nuclei come together and you have a normal, in air quotes again, amount of DNA. Okay, so M can be mitosis or meiosis. The lead in, what we call interphase, which accounts for G0, G1, G2, and S, not in that order. S is between. But that process leading up to division is called interphase. That looks exactly the same, regardless of what cell type you have. Okay, so it's really important. And again, we've talked about different cell types that undergo very different amounts of division, right? Some that almost never divide, some that divide frequently. And we need to figure out how this can happen, right? How is it possible for a stem cell to divide almost constantly, whereas a neuron does not do that, right? And it comes down to regulation. Okay. And specifically, we've got key checkpoints that we go through, and we will only proceed to the next phase of the cell cycle if we are ready and able to do so. Okay. And this is the key. So we have these proteins that are pretty much always present, right? We call them CDKs or cyclin dependent kinases. Think of this as like the lock on the gate between, let's say, G1 and S. Okay, you have a physical lock. The lock is always there. You can only get through that gate if you have the key. Okay, so we have specified gates or checkpoints that you cannot pass unless you have the key for it. Okay, and if you think of something that's in G0, it just doesn't have the keys. It's not going to proceed to that next phase of the cell cycle because it can't get past that barrier, right? In a different cell type, you might have the keys freely available, right? But we have these preset checkpoints. G1 leading into S is a pretty important one. We talk about that a lot. And the question is my DNA intact? Am I ready? Is it okay for me to replicate my DNA? Yes or no? Okay, we'll talk about some really important regulators of G1S in a minute, but G1S, is it okay if I replicate this DNA? Is this DNA okay? If so, if you have the key, which we call cyclins, we will only make the key or the cyclin if we're ready to go. Okay, great. Now we're past S. We've replicated our DNA. right? We're making our way, we're almost at M, right? Which is mitosis or meiosis. And we need to pause again, like, hold on. Am I ready? Was my DNA replicated correctly? Have I grown a sufficient amount for me to just divide my cell, right? Divide my nucleus via mitosis or meiosis, and then divide my cell via cytokinesis. Like, am I ready? We've reached the gate. Do I have the key? Yes or no? Do I have that appropriate cyclin that interacts with this CDK that will allow me to proceed? If no, shucks, go fix it, right? You cannot pass go unless you've got verification that you're ready to do so, okay? And there's also an M phase checkpoint where we want to make sure that we've aligned properly and we're ready to divide, because again, we don't want things like non-disjunction. We don't want weird chromosomal abnormalities when we go on to create daughter cells, right? So there are multiple checkpoints. The question is, am I allowed to proceed? Now, you can imagine when we talk about cancer, right? Cancer cells are characterized by typically abnormal growth and division. That's how you create things like tumors. These tend to bypass those gates when they shouldn't. That's the problem, right? And sometimes we mess with the gate itself. Sometimes you lose the thing that's preventing you from moving forward, right? If you knock down the gate, you could just walk right through. You can do whatever you want. Just divide, divide, divide. There's nothing stopping you. And sometimes there are mutations that accumulate that essentially give you the keys forever. The gates can still be there, but you can walk past them because you can unlock them. Okay, so there are two different types of genes that we talk about with cancer. Some that allow you to go through, or some that basically help you speed up, and some that prevent you from slowing down.
Interesting. I love how, because I love how you use like the gate, like the key and the door method. I always kind of thought about like a car, like the gas or the brakes.
I know, the
So
car
that's
is
the
the
classic
classic
one.
for
That's the
that.
classic one. Well, I'll
The
just give
car
my
is harder. The car is harder to connect to cell cycle checkpoints.
You're right. You're right. Cell cycle checkpoints are very much the door. If you're not thinking about cyclins, then it is very, very much easier. Because we have proteins, right? The ones that kind of cause you to give you the key are kind of the gas, right? These are kind of the proteins that kind of push you forward. Or you got brakes, right? And if you mess with the brakes, that means you don't stop. You just keep on going. And so these two classes are called our tumor suppressors and our proto-oncogenes. Now, tumor suppressors are our brakes, right? Because if you can imagine the term tumor suppressor means, hey, we kind of stop tumors from happening. We stop things bypassing gates. So those are the brakes. Proto-oncogenes are genes that aren't really a problem, but could be a problem. When you see the term proto, that means it's before. An onco is like cancer gene. It's like a cancer gene, where an oncogene, or like the gas, where it's like, hey, if there's a problem with your proto-oncogenes, which turn them into oncogenes, right? That's the gas. That's like, oh, I'm just busting through all the gates now. It doesn't really matter. And so understanding these two key kind of like molecular players, like the tumor suppressors and your proto-oncogenes, when there are issues with those, and accumulations of issues with those, that's when cancer happens. And actually, it's this idea of accumulation that what makes cancer so difficult to treat, right? You hear all the time, like the cure for cancer, but cancer is really like an umbrella term for like issues with cellular regulation. But like we just discussed, there are so many checkpoints, and there is a lot of different types of tumor suppressors and proto-oncogenes. Some that you might see, like a big tumor suppressor is P53.
Yeah.
To be familiar with that one. And that's a big molecular break, where if there's issues, P53, now you don't have breaks anymore. You just keep on going. A big, like a proto-oncogene, that actually I think we brought up, I don't know, have we brought up the HER gene, like HER gene
No,
before?
we talked about BRCA, but not her.
Yeah, BRCA is another tumor suppressor, but HER or RAS is another proto-oncogene as well. Again, you don't have to recognize RAS and HER. Really, like the main one is P53 to know, and that's a main tumor suppressor. There's a lot of what I'm trying to say is there's a lot of key players here where if there's a whole bunch of accumulation of mutations in many of these, but not all, you will get cancer. So that's why it's so hard to treat, because there's so many different causes of it that you would need to kind of target each cause individually in order to treat the specific cancer.
Yes.
Yeah.
Yeah. And that's the problem. Plus, And we've talked about what happens when you lose a checkpoint, right? You have abnormal division. When you have that once or twice, the problem becomes normally there are checkpoints to make sure, hey, did I divide my DNA properly? Did I replicate properly? Do I have any mutations? Let's pause before I divide again. Let's make sure I didn't accumulate mutations. Okay, as you bypass these checkpoints, because you've mutated a tumor suppressor gene, which is meant to slow you down, or mutated a proto-oncogene into an oncogene, which basically just allows you to speed up all the time, now we lose that pause and reflect step, which means you have one cell that goes too fast, right? That had already accumulated that original mutation. Now, the next time that cell goes to divide, which again, it's one of our hallmarks of cancer is uncontrolled growth. The next time it does this, it's probably accumulated even more mutations, but we're still not checking. Now it divides again, and we've accumulated even more mutations, and we stop checking. And now, and basically what we see is once we pass this kind of critical point, and a lot of cancers, by the way, use P53. I think P53 mutation is associated, I don't know the exact number, it's leaving me. It's like 70 or more percent of cancers involve a mutation in P53. It's that important. As you accumulate, they just get more and more complicated. And so the problem is, especially late stage cancer, even within the same type of cancer, the same originating cell between two people, their cancers can be incredibly genetically distinct.
Yeah.
Because by the time you catch it, they've accumulated so many random novel mutations that one treatment might work for one person and not the other. So it's incredibly heartbreaking. I wish that there was a one cure that could work for everybody, but because they're so genetically distinct, it's very difficult to treat, right? But oftentimes these cells, as they continue, again, unbelievable growth, which creates things like tumors. They typically also evade the normal messages that cells get when they behave erratically to undergo apoptosis, right? They don't undergo cell suicide, so to speak. Like they don't recognize I'm not doing the normal stuff. I should just call it quits so I don't hurt the body, right? I don't hurt the rest of the organism. They evade apoptosis. They also typically will recruit their own blood supply because uncontrolled growth requires a lot of energy, right? They recruit their own blood supply so that they can support their growth. And at times, parts of that tumor, right, those cancer cells can go into that blood supply and metastasize to another tissue, basically set up shop in a different, let's say, organ or a different tissue, and then begin growth of another tumor. So these are all enabled by a lack of cell cycle regulation. And again, we've got lots of very common mutations. We've talked about a lot of them. Even HPV is typically something that a lot of people are vaccinated against, right? But that is a virus that can actually inactivate P53, which we know can be incredibly cancer causing. That's a very important tumor suppressor gene. And ultimately it's very difficult to treat once you completely lose your brakes or once you really slam on the gas, which is what makes cancer so difficult to treat.
It's hard to get those like targeted treatments, like you said, because of those variants. And that's why a lot of treatments today, like actually historically, I mean, there's really cool research today, are actually very general, right? It's very much like on excising the tumor, right? Where like physically, like getting rid of it to do your best. Yeah. Or radiation. A lot of chemotherapy actually doesn't actually target tumors, but it targets rapidly replicating cells because tumors are under those. So it's like, oh, a lot of chemotherapies just target things that replicate rapidly, but that's why chemotherapies also have a lot of really serious side effects because like tumors aren't the only rapidly replicating thing in your body. So like a really, really common one is your hair cells. This is why chemotherapies usually are paired with a loss of hair because it's killing rapidly replicating cells like your hair cells.
Yeah. And your red blood cells,
And your
right?
red blood
Anemia is a
cells.
huge issue. Suppressed immunity because your white blood cells are also made by stem cells in your blood, in your bone marrow. Yeah, a lot of the side effects, even when you think of like the GI upset, the extreme symptoms you experience with chemotherapy are not because of the cancer, it's because of the chemo that is targeting any cell that divides rapidly, which includes the cancer, but also a lot of cells that are supposed to divide rapidly. Yeah, that's so tough.
Right. And I think, I mean, on that very, very happy note, like, I mean, I think that's a
I think the happy note is actually how much research is being done.
true,
Like we
truly.
have more advanced technology than ever before. I'm
Mhm.
particularly intrigued by CRISPR-Cas9,
Yeah.
which
Mhm.
we should mention because it's
It's funny.
awesome.
I've actually worked with CRISPR-Cas9 in my lab, like with the yeast. It was pretty cool. It's pretty cool. Well, it's a cool thing to mention because it has the ability to target regions of DNA and replace it. Which, if these are caused by mutations, the ability to do that is really cool. With CRISPR, there's a lot of like ethical and moral concerns with its use in
Mhm.
humans. But implication in things like genetic related illnesses is really, really intriguing and really like hopeful for the future. But still, a lot of research still needs to be done
Yes.
on it. Yeah.
Yes. But, at least if we're ending on a doom and gloom, there may be a light at the end of the
At the end
tunnel.
of the
I hope
tunnel,
so.
down
If
one.
anyone here loves research, it's a
Mhm.
great field to get
Great
into,
field.
right?
Awesome field.
We
Mhm.
need highly
Right.
advanced cancer treatments. And really it's going to come from
Mhm.
a really solid understanding of this material.
and
So
and amazing physicians like I'm sure everybody listening to will be one day.
yeah,
um
of
and so
course.
I think on that note, I think that's a really kind of great place to leave off on like DNA replication. We've hit on how DNA replication actually happens. We've talked about its regulation and the cell cycle and mutations. Next episode, we're going to be really shifting gears, aren't we, Molly?
Yeah, we
Yeah.
are. And I'm excited because
Mhm.
we had kind of been into this groove of like, okay, let's talk about how to tackle a particular section of the MCAT, and then let's give a high yield content thing within that. Like, this is something you might see in a passage like this. So we did Chem Phys, right? We did Biochem,
Mhm.
but then we kind of got off on a really, really high
Really
yield,
big
really
tangent.
important tangent. So
Yeah.
now that we've really told the story about how you understand DNA genetics and why it matters with gene expression and central dogma and all of these amazing things, now we're ready to get back to our rhythm, right? So
Hmm.
we're gonna pull a little skirt and we're gonna go into our final MCAT science section, which is psych soc,
Hmm.
which will be really, really cool. And
Hmm.
honestly, a lot of psych soc, of course, it's more about your individual psychology, how you interact with others, how societies function, etc. But keep in mind, our behavior, at least in part, is shaped by our biology, right? We
Hmm.
have the nature nurture debate, of course,
Hmm.
but like there is a solid amount of our behavior in our personality, in our emotional state, etc., that comes from our biology, right? Comes from the behavior of our cells and how they respond to changes in our DNA, etc. So it is gonna combine. I'm pretty excited about it. So Psych Soc is up next. Get
Right.
excited.
Get excited. I know I am. And while you're getting excited, check out all the amazing resources we have on our website. If you're looking for, because again, we've really focused on passages, or not on passages, on like the content here. But one big part that we haven't really, like, we'll really focus on next episode, but one I want to outline is passage practice with these content areas. And if you're looking for passage practice, we have so much free resources on our website. Go check them out. We got like thousands of questions. If you want to start practicing on DNA replication and mutations, go do some fundamental passages on those so you can see what we're talking about when we say things like cell cycle. When we talk about mutations and cancer, you will see these in your passages. So get some practice, and then we'll see you in the next episode. But until then, happy studying, everybody.
Bye, everyone.